Hello, this is Mr. Laudermouth. Good morning, good afternoon, or good evening to you as the case may be. This video is part 1 of my lecture on meiosis and sexual reproduction. So let's get started. First, we need to briefly review a little bit about the anatomy of the chromosome. A chromosome, of course, is condensed chromatin, condensed DNA wrapped around histone proteins. When you look at a chromosome, what you see usually is a shorter arm called the P arm and a longer arm called the Q arm. When a chromosome gets replicated in S phase of interphase, you have two sister chromatids attached at a region called the centromere, which I'm showing over here on the right in this picture. These sister chromatids would have identical genes. They're basically just clones of each other. But as we'll see in the next video, pairs of homologous chromosomes at this point can actually trade genes. Something to keep in mind, we have one chromosome here, but even after it's been replicated, this is still considered one whole chromosome. Yes, it's double-stranded, yes, it's got sister chromatids, but it's still considered one chromosome. That can lead to some interesting chromosome math. So if I've got one chromosome and another, of course I've got two chromosomes altogether. If I've got one replicated chromosome and another replicated chromosome, well, that's two chromosomes altogether, a total of 8 chromatids. Now, if I've got two replicated chromosomes and I separate the sister chromatids, I split the centromeres, I now have 4 chromosomes. So keep that in mind as we go through these steps. Let's start with a quick review of mitosis. This is a process for producing identical cells. The cells are just like the parent cell that produced them, and they're identical to each other. They're exact copies. They have the same amount of DNA, the same number of chromosomes, and exactly the same genetic information. Now, this process of asexual reproduction, reproducing simply by cloning cells by mitosis, some organisms, including eukaryotes, do this process. For instance, we have in the top right here an amoeba that's simply dividing by mitosis. That's how the amoeba population reproduces. We have here single-celled fungi, yeast, that reproduce this method. We call it budding. Even in animals, this is a hydra, a jellyfish relative. It reproduces by budding. Now this of course requires more than one round of mitosis, since the new hydra being produced is not one cell big, but it's still an asexual process. Going from one cell to two cells that are genetically the same. What are the disadvantages of asexual reproduction? What are the advantages? Well, disadvantage of asexual reproduction is that you don't really get any genetic recombination. We're never bringing in genes from another individual. That means the offspring are going to have pretty much exactly the same genotype as the parent, so they'll have exactly the same traits. Now, if the environment isn't changing, okay, those are probably good traits to have. After all, your parent survived long enough to reproduce and have you. But in a changing environment, having a collection of different genotypes in the population can better ensure survival of the species, because some individuals will have traits that are better adapted when conditions change. It's hard to build up that amount of variation if we're reproducing asexually. Well, what about us complex multicellular organisms like humans? What if we want to reproduce? Well, of course, we have specialized cells for that process called gametes or sex cells. In the case of humans, we're talking egg and sperm. Can't make egg or sperm by mitosis, because if those cells were identical to the individuals that made them, for starters, you might have figured out already, you'd end up with an offspring with double the number of chromosomes it should have. The egg had 46. And the sperm had 46 chromosomes, that's going to result in a zygote, a fertilized egg, of 92. If we continue that process generation after generation, we keep doubling the number of chromosomes. That doesn't seem like a very sensible way to reproduce. So if mitosis is not going to work for us multicellular organisms, if that's not going to help us to create offspring with the same number of chromosomes as we started with, as we can see in this karyotype here, we should have 23 pairs of chromosomes, 22 pairs of autosomes, and a pair of sex chromosomes. Well, if we're going to be successful at that, we're going to need a different reproductive process, a different type of cell division. Here's a karyotype for a normal male. Still 46, but this time the sex chromosomes, instead of being XX, are XY. Before we get into meiosis and how sexual reproduction is carried out by multicelled organisms, let's take a minute and talk about the fact that you get one chromosome of each pair from your mom and one from your dad. The fact that we can partner up these chromosomes based on the size of them and the types of genes they carry means that they have some things in common. Each pair of chromosomes is homologous. Now, that does not necessarily mean that they have identical DNA. It's just that they carry the same genes. So a pair of homologous chromosomes, for instance, might both carry the gene for eye color. Now, one could have the version of the gene for blue, the blue allele. One could have the version for brown, the brown allele, but they still both have the gene for eye color. It's the same information, but it doesn't have to be identical DNA. In the case of this very simple cell that's got 4 chromosomes, we can clearly partner up the chromosomes based on their size. We would have one large homologous pair, which could be replicated to form sister chromatids. At that point, then, we need to ask ourselves, once we've copied all of our chromosomes, once we've made those sister chromatids, how do we get then to egg and sperm with the correct number? Well, we've got to take that starting number 46 and somehow cut it in half. We want reproductive cells with only 23 chromosomes. That way, when the egg and sperm combine, 23 plus 23 gets us back to our normal diploid number of 46. So we need an egg cell with 23 chromosomes, a sperm cell with 23 chromosomes. So we've got to make gametes with half the normal number of chromosomes, gametes that are haploid, even though in both cases we are starting with a diploid cell, one with 46 chromosomes. There is a process for doing that, and it's called meiosis. At fertilization, the two combine, and we're back to an organism with 46 chromosomes. We call that fertilized egg the zygote. Okay. So if we're going to make gametes that have a reduced number of chromosomes, that have half the normal number, what it really means is that we are alternating stages in the life cycle of the organism between haploid and diploid. We go from diploid to haploid, from 2n to n number of chromosomes, when we do meiosis. This is the process that makes egg and sperm, and that is what's happening here. We're going from 46 to 23. Haploid gametes. Once we've made those gametes, they can combine by fertilization, restoring the chromosome number back to our diploid number of 46, back to 2n. N plus n makes 2n. So haploid, diploid. Now, of course, the haploid part of this life cycle we don't think about much because these are microscopic cells, but they are vital. That is how our species and other multicelled organisms tend to reproduce in both the animal and plant kingdoms. So each of these gametes has just one copy of each chromosome. They're haploid, 1n or n. When they combine, you might have noticed already, we're going to have homologous partners again. 2 copies of each chromosome, so we're back to a diploid cell, 2n. When that cell then divides by meiosis, we'll be back to haploid cells again. But notice something. Meiosis has some randomness built into it. We don't have to make gametes that are just like the gametes that we started with. We can have some recombining of chromosomes. This will result in gametes with a genotype different from the original parents, explaining why, for instance, parents and offspring are not identical. Yes, they share many of the same genes and chromosomes, but they can be recombined. Next generation, we're mixing things up. Because of natural selection, we have now the variation that natural selection can work on. It's a good thing. We often call meiosis a reduction division because while we are dividing the cell, the purpose is to reduce the number of chromosomes. We want to cut the number in half, go from diploid to haploid. So this process will make the gametes, getting us egg and sperm. Important thing to keep in mind. In the next video, we'll go over more of the steps of meiosis, but meiosis evolved from mitosis. Mitosis came first. So the stages and the parts needed to accomplish it are very much the same. But they are two totally different processes with two totally different purposes. Mitosis makes identical cells needed during growth, development, or to repair injury. Meiosis is all about reproducing a new generation. We want to make cells with half the number of chromosomes so that we can combine chromosomes in the next generation. Be careful you don't confuse the two. The names are quite similar, the steps are quite similar, but very different outcomes. So let's wrap up this video with a quick overview of meiosis. There is an interphase where, of course, the chromosomes get replicated. There's a prophase 1, a metaphase 1, an anaphase 1. Anaphase 1, the homologous chromosome pairs separate. Telophase 1 ends with cytokinesis. At this point, we have gone from a diploid to a haploid cell because of the way we count chromosomes, remember. We then go straight into a prophase 2, a metaphase 2, anaphase 2, where now the sister chromatids separate. Finally, a telophase 2 and cytokinesis that will get us 4 haploid cells. These steps in part 2 of meiosis are pretty much the same steps that occur in mitosis. If you compared these prophase, metaphase, anaphase, telophase 2 steps to your mitosis wheel, you'd see that the pictures are almost identical. But up here, this process, much different. This was our reduction division, cutting the number of chromosomes in half. So we do IP-MAT followed by another PMAT, PMAT. Notice that there was only one interphase. It's a good thing, too. We wouldn't want to copy the chromosomes again in between meiosis 1 and meiosis 2. If we did, we'd have canceled our work. We'd be back to a diploid number. We don't want that. We want haploid cells. Okay. So to kind of reiterate here, we've got an interphase where the DNA gets copied. We have a first part of meiosis in which homologous pairs separate. That gets us a reduction division, gets us to haploid cells. And then a second round of cell division where sister chromatids are pulled apart, getting us 4 haploid cells. Okay. Last slide for this video, we're going to prepare for meiosis. Again, back in interphase, we're going to duplicate our DNA. So in this particular instance, we have 6 single-stranded chromosomes. Our 2n number is 6. After performing interphase, we'll have 6 double-stranded chromosomes. Each one now consists of 2 sister chromatids. Since meiosis evolved after mitosis, that's why we still go through this process of copying the chromosomes. You might think it would save time just to start with 46 and just immediately split to 23. That would be kind of the efficient way to do it, but that's not how it happens. And because it's not how it happens, we have to go through 2 rounds of meiosis. So interphase, S phase, just like in mitosis, the DNA gets copied. At that point, we're ready to begin meiosis 1 with prophase 1. All right, we'll pick it up from here in our next video. Thanks so much.